Hi everyone, my name is Mike Clements. Unfortunately, Austin won't be able to join us today, so I'll present on his behalf. I'd like to begin by thanking the KSBNS conference organizers, Professor Jin-So Hsu and Rhino Bio for inviting me to speak today. The title of my talk is Functional Characterization of Complex Neural Models, New Tools for Organoids, Spheroids, and Organ-on-a-Chip Platforms. Organoids are transforming neuroscience, but to unlock their full potential, you'll need the right tools. Here, I'll discuss how Axiom Biosystems is optimizing the Maestro MEA platform so your lab can seamlessly incorporate these new cell models into your research. My name is Mike Clements, and I'm a Senior Vice President at Axiom Biosystems. I'm a neuroscientist by training with experience manufacturing stem cell-derived models in industry. Neural organoids are three-dimensional, self-organizing cellular models generated from human pluripotent stem cells, or primary tissues. Because these advanced 3D models can recapitulate many features of the human brain in vitro, neuroscientists are increasingly relying on organoids to investigate neural development, disease modeling, and drug discovery. Unlike Maestro MEA, many assay tools on the market today were not designed for use with 3D cellular models. Maestro MEA helps you go beyond traditional morphology and marker expression to recapitulate human biology and capture complex neural activity in vitro. With over 85 peer-reviewed publications using Maestro MEA and organoids, our platform is trusted by researchers studying a wide range of neurological disorders, including Alzheimer's, ALS, autism, epilepsy, glioblastoma, and Parkinson's disease. What is MEA technology? Multi-electrode arrays measure extracellular electrical activity. When excitable cells like neurons or cardiomyocytes are cultured on an MEA, microelectrodes embedded in the culture surface of the plate detect their firing in real time. Non-invasive measurements monitor activity for the entire culture. Organoids are a powerful new cell model, but there are two major challenges. They can be difficult to produce consistently. They can be difficult to study in a non-destructive manner. You've spent months making your cell model, so the last thing you want to do is fix and stain it to study it. At Axiom Biosystems, our goal is to help you capture the complex biology of your cells non-invasively in real time. This is our Omni Live Cell Imager, a simple-to-use whole vessel imager that can sit on a shelf in your incubator. It works like a flatbed scanner, imaging the entire multi-well plate or flask. The major applications for the system are for optimizing cell line development, testing different media formulations, monitoring cell growths, amongst others. Why image the entire culture surface? Well, when you do, you get a more accurate understanding of your cell culture. Here you can see the differences in cell coverage if you focus on just a small sample area versus the whole well. And in 2023, we launched a new 12-plate version of the Omni system called the Omni Pro 12. It has a robotic arm to shuttle the plates down onto the imaging surface and then back into the plate hotel. The experimental scheduler automates this process, reducing the amount of time you need to spend monitoring the progress of your cell cultures. Now I'm going to quickly cover how the Omni imaging system is being used to help standardize and make the production of stem cell lines and organoids more consistent. Cell line development can be challenging. For example, confirming that you have dispensed a single cell in a well, then tracking the speed of proliferation of this colony over time. As you can see, the full vessel scan of the Omni makes this effortless. We visit many labs where this process is still performed manually on a benchtop microscope. Prolonged periods out of the incubator is suboptimal for stem cells. It's also probably not the most effective use of your time. Here you can see an Omni full plate scan of a 6-well plate. Just like Google Earth, but for your cell culture, you can zoom down to reveal all the colonies in your well. Sliding the scroll bar shows you how these colonies grow over time. And the software will calculate how many colonies are in each well for you, so no more manual counting. The Omni can detect all the colonies in a well, count them, and track them over time. The quality of your stem cell culture going into your differentiation will have a major impact on the quality of the resulting cells. Monitoring stem cell culture proliferation can be very time consuming and subjective. Are your cultures growing as fast as previous cultures? Is the culture at 50 or 65% confluence? Will I need to passage the cells on Sunday, or can I do this on Monday? The new iPSC module on the Omni takes all the guesswork off the table, helping you to improve the consistency of your iPSC culture. The organoid module on the Omni can help standardize the embryoid body formation and expansion process. This can be a critical step in the organoid production process. Here you can see how the software can help you track critical features of embryoid body development, including diameter, roundness, and the number in the well. So I've discussed how Axion is providing tools to help you make more consistent stem cells and organoids. Now I'm going to cover how we're giving researchers deeper insights into their functionality of their neural organoids without disrupting them, using the Maestro MEA system. Why is there so much interest in recording from neural organoids on Maestro MEA? This seminal paper from Kleber Trujillo in Professor Alison Watry's lab demonstrated the complex neural network activity that develops in neural organoids. After 4 months in culture, nested oscillations were detectable in the neural burst firing, which mimicked the types of activity seen in the EEG from the developing human brain. What they also showed was that the same complex neural activity was not present when the same cells were used to make neurospheres or 2D monolayers. It was only present in the self-assembling neural organoids. At Axiom Biosystems, we're pushing the boundaries of what MEA technology can do, designing tools specifically for the demands of 3D biology. To that aim, we've developed 3 specialized MEA plates. 1. SpheroHD, for smaller neural organoids demanding high-density electrodes. Organoids aren't one size fits all. To accommodate smaller organoids, that is, approximately 200 micrometers in diameter, we've developed SpheroHD MEA, which features electrodes spaced just 50 micrometers apart. This ensures more recording sites per electrode to better map network activity in these smaller models. 2. Spheroguide MEA plate, for consistent, reliable organoid placement in a 48-well format. Positioning organoids can be a challenge. To make neural organoid recordings on Maestro MEA even easier, we've developed the Spheroguide MEA plate. The well design funnels the organoid over the recording electrodes in the plate, enabling faster and more consistent loading of the plate. 3. 3D map, our first MEA to feature flexible electrodes that wrap around your organoid to maximize contact across the surface without distorting its shape. 2D measurement tools weren't built for 3D models. No matter how many electrodes you have, you can only measure so much of a 3D sphere against a 2D plane. By integrating flexible electrodes, our innovative 3D map MEA can wrap around your organoid, allowing you to map activity across a larger area of the organoid surface. These innovations expand Maestro's capabilities, enabling high-quality, reproducible recordings across diverse organoid models. We can confirm that the organoid is touching the electrodes using the impedance mode on the Maestro system, and then switch to neural activity mode for the electrical activity recordings. Here is a video showing how we can confirm, using impedance, that the organoid is touching the electrode array in the well. With the neural module, we can then detect the spontaneous electrical activity from the organoid and the complex burst firing patterns. As the Maestro MEA plates are transparent, when combined with our Omni imaging system, we can seamlessly multiplex the imaging and functional data, so you have the most complete understanding of your neural organoid. We call this Connected Lab. Finally, with the need for even higher throughput, our customers are starting to automate their Maestro MEA assays. Here is an example from Revity, where they've integrated the Maestro Pro system with their PlateWorks 6.30 scheduler software. This integration enables automated recordings and workflows with liquid handlers and other instruments. So in summary, At Axion, we want to provide you with the tools to help you make more consistent stem cells and organoids, and then give you deeper insights into their functionality without disrupting them. As a point to leave you on, organ-on-a-chip models provide another approach for creating more physiologically relevant in vitro cell models. Axion has recently partnered with the microfluidic experts at Nectri to produce the first multi-well microfluidic plate with MEA electrodes integrated into it. Here we can see neurons seated in one compartment project neurites out and across into the neighbouring compartment. The electrical activity in the microfluidic chip can be recorded using the Maestro Pro MEA system. At Axion Biosystems, we're driving the future of organoid research. Maestro MEA is more than a system. It's a platform evolving to meet the complex needs of today's science. Join the researchers already using Maestro MEA to illuminate how organoids grow, function, and respond, and see how our advanced organoid tools can accelerate your research. I hope you're all enjoying the KSBNS 2025 conference. If you have any questions about the work I presented today, please contact the RhinoBio team or visit axionbio.com to learn more. Thank you.